Okay. So thanks, everybody. Great thing about being the last speaker is I can talk as long as I want. And if I want to take session and Mike for inviting me to give a talk, I'm probably the only non-true card-carrying geneticist that presented it. I want to tell you about the MGI-MEND collection, which some of you know about, but most people probably don't. It's a sub-collection that we started about 8 years ago now. Specifically, actually, Mike South and Gonzalo encouraged us to do this. I came with them with the idea, and they said yes. And so, our motivation is because most of the genetic collections is just taken from the pre-op areas of the surgical speech, which is for people with mentioned for cancer and certain diseases. And so. We thought that collecting samples from a common disease would be informative. So we thought that we could use this to utilize this collection to add to other ongoing collections for type 2 diabetes and type 1 diabetes risk. We thought there's potential for physiological studies on individuals with novel variants. We have a protocol for callbacks. If we find novel variants that can do deep phenotyping in the individuals, they Heidi for Alta was skip through this probably a year and a half. It's like us to get through the IRBs. Say we don't know what tests we're going to do, but we're actually going to do it. And if they cut it off, I'm doing repeated classes of the liver. I'm doing that. We also wanted to collect biospecimens for further study. Come back to that a little bit more. We also provide resource recruitment of subjects to related studies. And this actually is what I've been doing. Collection is being led by Ming Tan. Heidi, again, I mentioned her a lot because she actually helps me a lot in my life. Works with the Tom Institute, but she actually has been working with Ming to get this database functional. And we're actually taking advantage. I don't think is an important point of this database is that being co-collected with the diabetes registry. It's been ongoing in the endocrine clinic at the University of Michigan for many years. And so what we have with the diabetes registry is matched in a good phenotyping associated with these individuals. And these phenotyping well enough to really differentiate between type 1 and type 2 diabetes, which I'm going to come back to in a little bit. But also the medications or all that kind of stuff. It's much more easy to do this. So having a focused collection actually can help some of the things that we've been talking about today. So the collectors, we've actually done this. It's actually fairly effectively. We hire gap year students who want to get into medical school, and I've highlighted all the kids that have not been in medical school but are working with us for a year. So for students that want to take a gap year, get into medical school, come work with us. And so every year we have a new one, and this will be going on for about 8 years. All right, so how much have we got? We actually looks like we have about 5% of the collection in the MGI collection now. The types of diabetes are here. It's obviously it's more biased to type 1 and type 2 diabetes, about 2 to 1. Type 2 diabetes versus type 1 diabetes, and there's a smattering of others. We also collect from our obesity clinic, which we created a number of years ago with Amy Rothberg. And so we try to systematically collect from people who are undergoing medical weight loss program. We don't have as much on the other. Now, one of the things that I've been trying to get MGI to do for a long time is actually collect plasma, which is what we do in our collections. We actually taking the time that when we collect a tube of DNA, we actually spin it, collect the plasma. And actually, this plasma has actually turned out to be a popular thing for a lot of people. They've been used for a lot of controls, but also to specific studies that's going on. So about 4,300 samples that we collect. Now I want to show you a couple of studies, just real quickly, about how we have contributed to some very interesting studies through the collection. The first one is a study we did with what we're doing, detection ongoing with Rhythm Pharmaceuticals. So Rhythm Pharmaceuticals is a company that has a drug that it's septolanitide. which is an agonist for the MC4 receptor, which is actually one of the very distal receptors at a control speeding behavior. And individuals with mutations in the MC4 receptor are obese. It's probably 5 to 10% of morbid obesity in the country. So people with BMIs above 40 or 45 have a high prevalence of this of the low variance within the MC4 receptor that decreases signaling. And so this drug is directed at this receptor. Now, this drug is now approved for some rare cases of hormones or low variants or mutations in specific genes that signal up through the MC4 receptor. Notably, the leptin signaling signals up through MC4 receptor. POMC, which actually breaks down, it has mutations and there's not active processing of the POMC gene, and this causes a decrease in stimulation of the MC4 receptor. And there's numerous other things. And this drug is miraculous. The kids who have BMIs of 40 at 10 years of age, they take this drug, and it massively decreases their weight. Kids with leptin receptor mutations, they can take leptin, but this drug also works very well in those kids who develop antibodies and have neutralizing antibodies to these drugs. So now I'm actually happy to meet an old friend who now works with pharmaceutical. I told him about the collection MGI, and he goes, "This is what we're looking for." So with the support of a number of people, we actually went through and looked at the distribution of the BMI measurement from all the MGI samples. What you can see here is that there, not surprisingly, there are approximately 1,000, about 1,200 people that we found, this is a couple of years ago now, in the MGI collection that were the BMI of over 40. And so this is Michigan. We're the fifth most obese state in the country, and we're a couple of cheeseburgers away from being there before. And so we're rich in a population of more obesity. But what Rhythm had found is that if you just take a random obese individual and treat them with their drug, it's not very effective. So the garden variety obesity is hot. But the thought is can we find individuals who have a high response rate? And so here you can't see it very well. In the left side of this screen, all has little circles there, oval, are purported genes that are upstream of the MC4 receptor signal. And what they decided to do is try and find coding region variants in these genes that could result in decreased signal. And by identifying these individuals, enroll them in a study, or multiple studies in this case, to understand if the drug is effective. And so we got together with our great clinical endocrinologist, Elif Oral, who's very interested in rare variants, but now with a lot of the other people with the diabetes clinic. So we identified those very obese individuals from the MGI database, but also, which is obviously rich because of our obesity clinic and the type 2 diabetes that we have. And we found a significant number of people, I forget what the number is now, but over 30 people who have potentially actionable allelic variants that decrease the signaling up to the MC4 receptor. And these individuals are being now enrolled in this clinical trials. Interestingly, in this, I wanted to alert people to, Rhythm actually paid for whole genome sequencing on 1,600 of these, all these samples that we sampled, 1,600 people. So somebody who's interested in that subset of samples, we have a lot. And this is a way that it's done. I think this is a really interesting way to do it. And this is probably the way these clinical studies should be done. In our biobank DNAs, after the whole genome sequencing, variant IDs were called. There was SAGO confirmation. This is all done in de-identified data. And then when we identified individuals that are associated that had these allelic variants, they actually go through another sequencing via certified manner to get them. And so the interested individuals. Now this is an ongoing study, but this is the kind of thing I think we're all interested in doing, actually find actionable and bullet areas. And I think we're probably close to being the first people who actually done this sort of approach to look for actual variants and actually tap a drug and treat these individuals. I wish I had great results. The study's ongoing, but I know not from our site, but from some other sites that I had talked to, that these drugs do look like they're working into some of these individuals. So it's actually pretty exciting. Although it's an open label trial, it's hard to do a case control study. Although we're trying to convince them to take a random sample of people without the variants. Or multiple variants to see how they respond to this medication. So these are all the genes that are predicted to be upstream that signaling to the MC4 receptor, and the enrollment criteria are shown here. And this is one of the studies that I'm on to study, the MGI-MEND study, which we're doing here at the University of Michigan, along with another thing that called study. So it's setmelanotide once daily for 52 weeks versus a placebo, and seeing the response in these individuals. We also have been doing a number of investigator-initiated studies with different people. I'm just going to present a couple of them. I think we've now, through all the samples, there's probably 10 studies going on with various parts of the collection, either genetic stuff or the plasma. One that for a postdoc of Mike's and Mike and I and Scott Solomon Core, along with a really bright postdoc, that showed looked at the evaluation of polygenic risk score to differentiate type 1 and type 2 diabetes. Not everybody thinks about type 1, type 2 diabetes. It's pretty easy. If you have type 1 diabetes, you're young, and you take insulin. Type 2 diabetes, you're old, obese, so you take insulin or you don't. The problem is that there's probably some misclassification. So when you look at the UK Biobank, there's a lot of people who are a lot older that seemingly probably are 80 years old and have type 1 diabetes. Type 1 diabetics, about if you're 80 years old, probably aren't alive for the most part. And so probably this misclassification. So we wanted to understand, and this came from an early study, a very small study that we did with this, that suggested that type 2 diabetes, there's an overrepresentation of type 2 diabetes genes in type 1 diabetics. The classification of type 1 type 2 diabetes is actually, it's probably some overlap in these diseases, depending upon the age of onset. So what Sarah did, beyond telling, did is she created a polygenic risk score from the known to low variants associated with type 1 and type 2 diabetes. What we found in this study is that the type 1 diabetes risk score in the UK Biobank could accurately classify type 1 diabetes. But the problem was, again, is that some type 2 diabetics give insulin. So we did age cutoffs for these individuals. But with the MGI-MEND collection, we have good phenotyping on the individuals and understand what type 1 and type 2 diabetes. And when we repeated this in the MGI-MEND cohort, actually it replicated very well. On the other hand, type 2 diabetes polygenic risk score could differentiate type 2 diabetics from controls, but they actually proved that they weren't statistically different between the type 1 and type 2 diabetics. So you could use type 2 diabetes risk score to differentiate a type 2 diabetic from type 1 diabetic. And this would go along with the suggestion that there are some enrichment of type 2 diabetes genes in type 1. And then the other things that we've been doing is actually contributing samples with the sorts of GWAS. The first paper we were on, this is now in preprint form. It's being submitted for some regular publications, and it's adding some more allelic variants to the number of people that are number of allelic variants that are associated with the risk of type 2 diabetes, expanding this polygenic risk. So that's a couple of things that we. The other thing, and Gonzalo's not here, but I want to thank Zalf. So about 6 years ago, I took over something called the Taubman Institute. And we've been funding a lot of human-related studies, both the longitudinal studies. Actually, Safi is one of our premier fundees, I guess. And we have 11 subjects. And one of the things that we're insisting on, or insisting on, insisted in, highly encouraging, is that these multi-investigator projects actually get consent for deposition into the MGI database, along with their biosamples going into our biorepository. We think it's really important to actually now have more focused collections of certain diseases, because then it's much more useful for the whole community to have at least a concentration of specific disease types, such as the vasculopathies at Southeast generating. We have a large project looking at psoriatic and other autoimmune skin diseases. So I think these are the other kinds of things that we're doing. And we have only about 500 samples collected from the studies we're actually doing. And that is my last one.
These were fantastic. Can we count for one or two questions?
That really is amazing work. So you suggested the search for additional variants. In addition to the leptin-related variant to differentiate type 1 and type 2 diabetes, you probably have considered the direction of adding various clinical scores, like relatively purely clinical scores, to the genetic score to come up with a combined score to accomplish the differentiation between the two types.
Yeah. So that absolutely, and if type 1 diabetes is probably, the phenotype is autoantibodies. The people that we diagnose with relatively thin individuals with quote type 1 diabetes at 30 years of age, a lot of those individuals could have something called LADA, latent autoimmune diabetes of adulthood. And only about 80% of those individuals are antibody positive, the typical antibodies. So one of the things that we're really interested in doing is actually using the type 1 diabetes risk score to identify those individuals as true LADAs, because you want to actually approach the treatment of that subset there. Unfortunately, people don't think about LADA a lot, and they don't test for the antibodies a lot. And so these kinds of this kind of approach may be helpful in those that are either antibody negative or that people have just not thought about the disease the right way. Conversely, I think the potential for an increased enrichment of type 2 diabetes genes in type 1 diabetics, I think, is really interesting. It could give us some idea of maybe why treatment different, why some individuals with type 1 diabetes actually look much more like type 2 diabetes, not just by body habits, but much more insulin resistance and things like that. So these are some of the reasons that I think this is going to be helpful.
That was really nice set of approaches that you were showing where you have for the rare variant, genotype positive and genotype negative species, then she had them become a pile. I'm curious, how scalable is an approach like that, to do some genotype driven, kind of molecular phenotyping, right? To figure which patients should be in this kind of study.
Well, so I think this is it actually shows the value of getting a genotype on everybody that comes into our hospital. I think talk about long QT intervals and things like that. It's hard. If you want to give an antibiotic, going back to the study, if you want to give an antibiotic, I'm not sure we're going to wait for a genotype to come out. We can give an antibiotic, but if that's actually present already, you can actually just pull it out of the database. And that's actually the kinds of things I think we probably need to fill folks. So you can actually do that. So, and so you see this, the lower variant POMC that actually causes decreased sibling. Perhaps since a woman has a BMI of 45, you could just treat her with this drug and have high confidence that they may respond, depending on the outcome of the clinical trial of this.